Schmidt shows up, whether those eels really go up to Sargasso Sea. That really happens.
We may present a little info on that today. Excellent foreshadowing.
Okay. So we have actually a team of folks that have been working on eels, research end, on the education end, and we're kind of excited to bring a lot of the work that's been going on together. Karin Limburg is going to be the first star of the evening, and I think a lot of folks know Karin. That's probably why the crowd was as big as it is. We personally here at the Hudson River Foundation known Karin for quite some time. Karin's one of the few people that has gone through some of our various programs here at the foundation, from a Polgar Fellow to a Graduate Fellow to the recipient of several Hudson River Foundation grants. And it's one of the things that we think is a real success of having an organization like the Hudson River Foundation that can support students that grow up to be adults and conduct.
Or not.
Or in the case of that turn into such productive scientists, and that they still maintain that love and affection and interest in the Hudson River, and we can benefit from the fine work that they do. So Karin is a professor at the Department of Environmental and Forest Biology at SUNY, Syracuse, SUNY College of Environmental Science and Forest. I wish they would change the name. So she's trekked down from Syracuse to chat with us today, and Karin is a fish ecologist. She loves anadromous fish. She worked on her PhD with shad, and continued that work. Worked on otoliths, worked on eels, worked on a variety of things, lampreys. And I guess what we worry about here in Hudson sometimes is that she gets these callings from overseas for her time on other species. And we want to make sure that she spends enough time here working on these very valuable things. I'm going to let Karin introduce her team members, but we're here for an interesting and exciting talks about American eels. Karin.
Thank you. Thank you very much. Everyone is high up there in webinar land too. If you can't hear me on the webinar, tell Sarah about that, and I'll try to stand closer to the mic. I'd like to first introduce my former master's student, Sarah Mount. Please stand up and say hello. Actually was involved with this project from the very start. We have a project that was funded by Hudson River Foundation, and Sarah, even before she became my student, actually was very interested and talked to Bob Schmidt and myself about it and helped us to write the grant. So she is very, very invested in this project, and now works back where she was before that at the Hudson River National Estuary Research Reserve with Mr. Christopher Bowser, and who is an educator. Also, both of them are educators. And Chris was a former educator on the Clearwater, if I remember right. He also was on Sarah's committee, and so he gets to continue. And now he works with her, so he continues to torture her. Now I've handed off the torture to Chris. So without further ado, then I will try to get this thing started. The hands that you see there are from another former master's student on a previous Hudson River eel study. So this is an outline of today's talk. So general, first what we're going to do in this talk is a little different than most of these presentations. We're going to kind of hand off the seminar back and forth. So first, I'm going to introduce you to the eel a little bit. Emphasis on make eels great again. Quick summary of some previous Hudson River eel studies, and then we're going to talk about highlights of our current studies, which sort of go back and forth between our funded work from Hudson River Foundation and what the state of New York is doing. through its glass eel program. And then we thought we'd wrap it up with sort of taking around to what's happened to eels in our minds. It's gone from just being a biological organism, not just any biological organism, but a special one, to a river icon that actually serves to bring people together around the river. So that, I think, is pretty important. So that's where we're going to end up. So freshwater eels are catadromous. You mentioned anadromous, but there's also the other way. So anadromy means that fish like salmon and shad and river herring use marine waters to feed and grow, but then they come up into freshwater to spawn and rear their young. And that's a very much of a simplification, but that's kind of the general idea. And catadromy is the other way, that fish that are spawned in marine waters move up into freshwater to live and grow. So the freshwater eels are a family, Anguillidae, which do this. We now know that they do a whole lot of other things, including live entirely at sea. But this is the general definition of catadromy. And so they have actually, yes.
Even stay at sea part, what is that?
That means that some eels don't migrate into freshwater at all.
Okay, so that's just the freshwater eels, not the shad.
It's sort of a misnomer.
Okay.
Eels haven't read the migration book. So, like many of these species, they haven't done it. So they just do what they do, pretty much. So they have a set of complex life stages, and we tend to like to give them names. So the larvae are specialized. So the larvae, as you're going to see, are born at sea, and they're specialized to drift. They actually almost look like willow leaves, and they're called leptocephali, which actually means narrow head, narrow heads. They transform into small eel-like creatures that you're going to see pictures of that are called glass eels because they're glass transparent. Then they develop the yellow-brown pigmentation, which is called the elver class and elver stage. When they get a little longer, sometimes they're called bootlace eels. Then yellow phase, as they get larger and become a little bit more yellow. Now, not all eels are yellow. There's a lot of color variation, but that's sort of the general name. And then finally, they mature into a phase called silver eel, which is not the actual final adult. It's sort of more like being a teenager. And as teens, they get restless and they move back to sea. So you're going to hear about that too. Now, eels were very mysterious, and we didn't know a lot about where they came from. And I could tell you a lot of things about what people used to think they were, like Socrates, Aristotle, I think, thought that they were formed when the hair off mare's tails would fall on the ground in the mud, and they would turn into eels. So that was one theory. But this gentleman, but this gentleman here, Johannes Schmidt, he happened to marry the daughter of the owner of the Carlsberg. Oh yeah, that's why I can't use the pointer. He happens to marry the daughter of the Carlsberg brewery magnate. And actually, Carlsberg funded a lot of expeditions and has had an intimate history with the International Council for the Exploration of the Seas, which is a large intergovernmental association of people who study the sea and fisheries therein. So Schmidt started off getting grants funded by Carlsberg, but from the Danish Queen, and went out in ships to collect fish, to collect actually eel larvae. And so as he went out, they started getting these leptocephali here. And so as they were, if you look at the bottom leptocephalus, that's the eel larva. It looks like a willow leaf. That's closer to Europe. And so as you went further and further, it would be like going up in the picture there. You get into smaller and smaller leptocephali. And finally, the smallest that they found was near the Bermuda Triangle in the Sargasso Sea. So this is a very mysterious part of the Atlantic. So there's this big gyre, and it's its own ecosystem there. And I'm going to not show you the distribution that he found of European eels, but just jump straight over to American eels. And so you can see that the further up, away from the spawning area, they're larger, and then they get smaller and smaller coming into the Sargasso. But what essentially happens, do you guys have a stick pointer or something like that? Something like that? I'd like to gesticulate. There's nothing here. Okay. Anyway, so, okay, I'll just, perfect. Thank you.
That's glass, by the way.
Oh, okay. Basically, the eels are riding the Gulf Stream, and as they come around, some will settle down here in northern South America, some will actually go up into the Gulf of Mexico, and some will ride around like this of the North American species and enter estuaries as they go. So again, here's what we see. This is a fully developed leptocephalus, and this is from a classic book written by a German gentleman. But as they mature, they actually shrink in size and get down to what looks like this is the glass eel, and then the small little elver phase there. So they swim up estuaries in droves, and you're going to hear a little bit more about this from Chris, some of the economic impacts of this. And they actually can climb vertical surfaces. They do a lot of amazing things, but so when they're small enough, they just adhere with capillary action to vertical surfaces, so they could actually wriggle their way up vertical surfaces, and they can get up some of the steepest waterfalls you can imagine. The yellow phase, which is really the growth phase, can live in a very wide variety of habitats, and they can also eat many, many different things. And I won't go into this morphological variation of the skulls, but their growth rates are also incredibly variable. So eels are, if you want to think of one word to associate with eel, variability. They're just incredibly variable. But eventually, what they try to do is accumulate enough fat to make this long, long trek back to the Sargasso Sea. And when they become ready, then they transform and become this next phase, which is called the silver eel phase, and their eyes get big because they actually go into great depths. They actually do very extreme vertical migration. And when they are fully silvered up, then they stop feeding and they just make this trip out, and we'll see a little bit of that. We also, I'm just bringing up a little index that's used, it's called the Pankhurst Index. It's just a way to characterize the shape and size of the eye relative to the head. It's a maturation index that Sarah will talk about. So just summarizing then this life history, we've got the eggs to the leptocephalus, to the glass eel, the elver and yellow eel to silver. And we do know that actually when they go out back to sea, enough work's been done on Japanese eels and a little bit of work also on American eels in vitro to tell us that they become basically egg delivery systems, and they just basically, once they go out there, they spawn and they die. And I think someday we're going to find the equivalent of the elephant graveyard down at the bottom of the Sargasso Sea, which has a real down there, I think. On this, I also show you the route that they take to go up into the Hudson. And I don't have time to talk about it today, but I actually think that they had a back way, a back door into the Hudson River watershed too, through the Mohawk. And if anybody's interested, I can talk to you about it afterwards. Just a bit on some of the early Hudson River and HRF funded eel studies. David Secor, a good friend and colleague at the University of Maryland, and his grad student Wendy Morrison studied eels in the main stem of the Hudson in the 1990s, and they actually caught the very beginning of an invasion of a parasite that infects the swim bladders, and we think that's a serious problem because although they're pretty sedentary in rivers and lakes and ponds, they do have to do very dramatic vertical migrations when they go out to sea for that big spawning migration, and so they need to be able to regulate with their buoyancy with the swim bladder, and if it's full of parasites, it's kind of tough to do that. So Wendy caught the beginning of that. My student Len caught sort of a little bit further on, and Sarah also worked on some of that in her master's work. But what I want to show you here is just, if we look at a plot of age versus the length of eels, what we can see is that there's just a tremendous amount, remember, variation in size at age. And so they grow in very, very, it's a very variable thing. But in particular, you can see that the triangles and the little circles here, which are the open ones, the George Washington Bridge and Haverstraw, that's down in the lower part of the estuary. And there's a lot that says that this part of the estuary is a very productive part. We can see that with many other studies that are done in the Hudson, but it really does seem to show up with eels. And I got these images from Wendy herself. These are cross-sections of little things called otoliths, or ear stones of fish. I'll talk a little bit more about that later, but they're part of the hearing and balance system inside the heads of teleost fishes. And I do a lot of work with these myself to look at things like age and growth. You can see these bands, these growth bands on them, like tree rings, and those are actually annual growth bands. And so what you can see here is the upper left corner, we can see that I think anybody can count these rings. I'd like to say even my husband, who's a biogeochemical modeler, can count these rings. And so you can see that the fish that was caught downriver at George Washington Bridge grew very, very quickly. It's a 49-centimeter fish, about 5 years old, versus a 45-centimeter smaller fish that I think has something like about, I usually have my class count the rings on this, but I think it's about 20, maybe this one might be 15 years old. I'd have to stop and count. But it's clearly a lot older. Yes?
How do you know the age?
We count these rings. So here we go. Sarah, where do we start? We start here, is that right?
Yeah, I want to do continental age.
So we can talk about continental age. So we usually start at a settlement mark, and then 1, 2, 3, 4, it looks like on this one. And again, here, I don't know if that's the ring that we start with, something like that. And 1, 2, 3, it can get a little tricky, 3, 4, and so on. And this is colored to emphasize the rings too. But it's a lot like tree rings, but it can actually be difficult sometimes to count those. These are nice samples. And then another study, this is actually my own first foray into Hudson River eel work. I'd done some eel work before. I worked at the University of Stockholm back in the 90s and did some work with colleagues there on European eel. And knowing how interesting they were, I convinced the Hudson River Foundation to sponsor us to do some work. And what we did was we tried to expand the work that Wendy and Dave did up into the tributaries of the Hudson and ask how important those are. And it turns out they are quite important. We actually published the highest densities of eels ever published in the lower part of a tributary called the Hanacroy, which is not at somewhere between New Baltimore and Albany. And this is my former student, Len Naku, with the electric fisher, and you can see Bob Schmidt behind him. Then I can't remember that undergrad's name, a Bard College student. But we characterized those eel populations and the demographics. And one of the things that we also did was quantified natural and artificial barriers in the system, and found that they did act as kind of filters on the population. So the further you go upstream and you encounter a barrier, if it's a waterfall, natural barrier, it takes a while for eels to get past that, but they can. We found eels in places up waterfalls that you just wouldn't believe. But then a vertical barrier like this on the Fosaic Creek in Newburgh to the right, that they can't pass. It's much shorter than many of the waterfalls we saw, but they cannot really get past those unless they're tiny glass eels, and even then it's kind of a stretch. Yes, they can swim out on a wet, rainy night and kind of crawl around there, but they don't do it so much. So we found that actually the artificial barriers are, in fact, a problem. And Bob Schmidt, our colleague who's not here today, has advocated strongly for putting means for eel passage into tribs to get them up into the further up. I won't go into reasons why, but it actually has a conservation biology implication to do that. And now I think I'm at the point where I'm going to turn this over to my esteemed colleague, Mr. Bowser.
Well, thank you, Karin. We'll see you again shortly. So I currently work for, my name is Chris Bowser, everybody calls me Bowser. I currently work for the New York State Department of Environmental Conservation's Hudson River Estuary Program, National Estuarine Research Reserve, in partnership with the Water Resource Institute at Cornell. But back in my Clearwater days, my introduction to eels was right around the corner. We were trawling aboard the Clearwater with a bunch of students at Bay Ridge Flats, which is a beautiful triangle of biodiversity just south of Governors Island. And we caught a toy recreational vehicle. And I was like, look at this toy recreational vehicle, which was covered with barnacles and hydroids and bryozoans and sea squirts. It was this beautiful artificial habitat unto itself. And I felt like the whole group was gripping, was just completely in my thrall. They were looking at this, and I was like, wow, I'm really talking about this great. They're loving this talk. And then unbeknownst to me, outside of the driver's window of the toy recreational vehicle, what they were really looking at was this eel that was slowly unwinding. They didn't care about bryozoans and blah, blah, blah. They were watching this eel unwind itself out of the window of the recreational vehicle. And I was completely flabbergasted and fascinated, and I started to look into this beautiful, beautiful animal, the American eel. It was my introduction, and I haven't given up since. And one of the things about eels is that's an important thing to know is that the 20th century and the early 21st century has not been kind to the American eel or really the group of freshwater eels worldwide. And because this animal lives in so many different habitats, oceans, estuaries, watersheds, uplands, lowlands, anything that is a driver of environmental stress is going to affect eels. Climate change, check. Habitat loss, check. Dams and barriers to migration, check. Boy, oh boy, eels have had a tough time. Particularly when you look at the Japanese eel and the European eel. And one thing that we haven't mentioned is the fact that people like to eat these animals. But they're not always hard to catch. If you think of the sort of a diagram that sort of represents the eel life cycle, what you end up with is a bit of an hourglass shape, where you've got the big wide ocean of salt water, and then the big wide watershed of fresh water. But those eels have to go through a little choke point at some point in their life, and we call that choke point an estuary. So during that life cycle of eels, where they're spread out through the ocean, they're spread out through the watershed, many of them funnel in through estuaries. And at that point where they're funneling in through estuaries, they're at that glass eel stage. And even better, that glass eel stage is fairly predictable in time and place. So the glass eel season here in New York is roughly February through May. It's usually even more contracted than that, but it shifts from year to year. So this is a time when you can catch glass eels with fairly good predictions that you'll catch something. And that becomes a great way to study eels because, okay, you know that you're going to have a bit of a concentration you can rely on. But that also means that if you're interested in eating eels, that also becomes an interesting stage in their life. And some of you may have read, yes, Mr. Jackman, Dr. Jackman.
I've got a question. It's the eels, the glass eels, we all know the paradigm that they head upstream. Why would they leave the most productive part of the river to mature? Why wouldn't they want to stay there? Which is contrary to other anadromous fish. They move down.
Sure. Three potential answers. One, not all of them move. Two, it could be distributing themselves. Sure, it's productive, but there's also a lot of glass eels, so you need to pioneer out there and find better habitats. Three, great question, and we don't really completely understand the answer.
slide with the Seacorn and Morrison to show the highest growth rate in the lower estuary. So, you know, you would say, why leave when the groceries are here?
It may be because there's a lot of eels that are trying to capitalize on those groups.
But if they're all getting big, you're taking a risky chance. You're moving to California.
You absolutely are.
You guys are a second visualization company.
Well, here's one thing though, very, very often we find the largest eels further upstream where they've had that chance to sort of monopolize a smaller, but at least monopolized food source. Monopolize might be overstating it, but that's often where we find the biggest yellow eels near adult eels. Good question, Al. Up in the state of Maine, there is a legal glass eel fishery, and because the Japanese eel and European eel stocks have so dwindled in the past 30 years, it has been a cowboy mentality of skyrocketing prices of up to over $2,000 a pound every spring. And that becomes just a sort of a major story about the eels as a commodity. In fact, if you go to a sushi restaurant, even here in the city, there's a good chance that the eel you're eating was hatched near Puerto Rico, caught in Maine, flown to China, grown in farm ponds throughout China, then flown back to the United States. It's an amazing life cycle, and you can find eel products. Here's Karin brought this today. Here's a little roast eel in red yeast sauce. I'm sure we could go down the street and find some unagi in a restaurant. So there is a commodity aspect to this animal that people are interested in part because of the decline of eels, in part because of this interest in eels as a commodity. There has been also, luckily, an increased focus on eels as a topic of research. There's a group called the Atlantic States Marine Fisheries Commission, the ASMFC. I highly recommend you check out their website. And basically, the states that are a part of this, from Florida to Maine, each of those states has some level of glass eel monitoring that they are doing on an annual basis to keep an eye on how eels are doing along the east coast. That is they started doing this in the early 2000s, and that was a very important piece of this story because we ended up with a couple of great models of eel research to follow. First of all, New York State's sort of sentinel site for eel research is out on Long Island Sound, and a special shout out goes to Carol Hoffman and Jesse Hornstein and the DEC crew out there that has been steadily monitoring glass eels with nets in the mouth of the Ames River for many, many years. Related to that, in the early 2000s, our buddies Bob Schmidt and Tom Lake took fight nets that looked very similar to this net right here, and they started to sample a couple of tributaries in the Hudson River estuary, using techniques based on what Carol and the ASMFC have protocoled out. Back in 2008, my supervisor, Steve Stan, basically came to me and said, Bowser, look what they're doing, look what the DEC is doing on Long Island. Look what Tom and Bob did in the early 2000s. Why can't we do this, or a version of this, with student volunteers and trained adults and watershed groups? And thus started what we call the Hudson River Eel Project. It's a community science or a citizen science project. And basically what we're doing is ASMFC-based protocols at sites from Staten Island all the way up to near Albany. And at all of these sites, each spring, we set up these spike nets, these triangle-shaped spike nets that are basically set up where tributaries meet tidewater. Some of them are in more saltier places like Staten Island. Some of them are in fresher water places like Bard College. And these nets are basically facing the estuary. So they're not catching the water that's coming downstream. They're catching the glass eels that are swimming from the estuary up into these tributaries. So we're trying to get some sort of metric every year of the glass eels that are coming into these tributaries. Each day, trained volunteers. Some of them are college students or high school students. Some of them are adults and watershed groups. Some of them are scientists that work with us. These trained volunteers go out, they open up the nets, they take those eels out, both the glass eels and the slightly bigger elders. They count them, collect some environmental data, and then take those eels and release them above the next barrier to migration. A, to help them, get a fin up on their migration, but also because we don't want to catch them again. So get out of here. We don't want you back in our net again. And we have been doing this at some of our sites. This is year 10 of doing that. So we're really happy about that. And since we've started, we now have, this year, I think it's probably going to be well over 600 volunteers a year that are helping us at 12 or more sites. And last year, our cumulative total was 450,000 glass eels and elders caught. We highly suspect that this year, probably today, as we're speaking, somebody in a net in Poughkeepsie is going to pick out and count the 500,000 glass eel that we've caught and released above the barrier. We don't know when, but we'll figure that out. Or if there's ever a press release, we'll pick a day and call it that. So that's sort of a nice big picture, but that we want to delve a little bit deeper into that information about these glass eels and elders caught. So when we look over 9 years of data, and we look at the total glass eels caught at all sites, at first glance you can say, wow, look, it looks like the eels are doing great glass eel wise. But this is a little misleading because we've added sites over time. So let's do this instead. Let's say we'll add up all the eels caught at all the sites and sort of divide out a catch per unit effort that we'll roughly call eels caught per day per year. So the total number of eels caught divided by the total number of days times site sample. And we still see this sort of upward trend where over time it's not a linear growth. There's some bumps in there, but it looks like the first few years of sampling we were catching far fewer eels than the last few years of sampling. But again, when you look at a big picture like that, all right, but let's dive in a little bit deep. So let's look at two sites. Let's pick two sites. One of them is Furnace Brook, and this is in northern Westchester County near Croton Point. And at this site, what's interesting about this site is our average number of eels caught per day hasn't varied all that much. Here's one example where the variation doesn't seem to be that great. We seem to be fairly consistent in the average number of eels that we catch a day there. And it's been going along, and so there's been some ups and downs. But then let's compare that range of variability to what we see up in Poughkeepsie at the Fallkill Creek. And what we see there is this very distinct range of variability, especially when you compare that range of variability to what we had at Furnace Brook. So there's Furnace Brook's range, fairly steady, especially when you compare this to what's going on upriver. And we've got some hints that this trend is repeated at other upriver sites as well, like Passaic Creek and Black Creek. There seems to be a real difference in that range of variability downriver versus upriver. Why that is? Well, we can talk about that and speculate on that a little bit. There's a couple theories, but they're just the hypotheses that we're trying to work our minds around. This data also has provided us with some other information. So, for example, in 2014, we saw something really interesting. We didn't see sort of a rise and fall with the tides or anything else. It seemed to be almost a stampede of eels. One big group of eels seemed to be going up the river and peeling off into tributaries as they went. So we saw these peaks, and the magnitude of the peaks changed. Down at Staten Island, we saw a peak on April 14th, and then on April 21, at Furnace Brook, we saw another peak. Even though it was a smaller peak, it was still a distinct peak, and we went up the river, and again, there was this really solid peak, and as we went up, we realized, hey, we can kind of, if that really is a stampede of eels right up, we kind of look at the speed that these glass eels are taking. So when you average it out, downriver, these glass eels are traveling about almost 11 kilometers a day, which for a little 2-inch glass eel, that's booking it. Now, probably they're probably using tide and current to their advantage, but remember, half the time tide and current is a disadvantage for them. So that's kind of impressive. And they are distinctly getting slower as they head upstream, which may not be a surprise. It's a lot of work to go up the Hudson River, and we see that reflected in their speed, their kilometers per day. So there's some interesting little stories that we start to tease out of this data as we look at this glass eel project. There's a couple other devices, by the way, that some of you I'll just mention because they might be of interest to some folks. Putting a fight net in a creek is a lot of work. It has to be checked every day. You need a very special kind of site. Sort of our entry-level eeling is something we call eel mops. And this is basically, literally, you take some polypropylene line and you unravel it all and stick it to a cinder block or something like that, and you bathe yourself a little island of artificial habitat. And it may not be as strong on the quantitative data, but it's great for presence, absence, timing of eels. All right, as we know from our Yonkers experience, Bob Walker's here, everybody, who helps us out. He's basically one of our early warning systems, okay? Down at the science bars, they're doing a great job of putting in eel mops in February and letting us know when, ta-da, eels are in Yonkers, everybody start rounding up the troops. So we got to get the nets in. Another thing that we work on is, okay, we've got this glass eel thing not down, but we're taking a look at glass eels. What about the next stage? What about those barriers? Great, you put plastic bags of eels over barriers. What about the rest of them you're not catching? And there is a growing movement throughout the Northeast United States. It stutters in some states and advances in others and so forth, to try to think about fish passage over dams. And one of the things that we use is sort of low-tech, low-key, almost homemade eel ladders, or what we like to call eel evaders. And this is a very, very simple device. Basically, you've got a bucket and a long tube, and that tube is packed with old netting or some sort of structure. And you get a couple of hoses at the top of a dam, and you bring them on down so you can get a nice little flow going down this tube. And ideally, your eels will sniff or feel out that attraction. They want that flow, and they'll wiggle their way on up through that ladder of old net, plop down in that bucket, and twice a week, volunteers can empty the eels out of that bucket and get them above dams. And what we found, and I think this is reflected even in more highly engineered eel ladders, is that the sweet spot for eels that are trying to ascend dams seems to be this elver stage. Not this small glass eel stage, and not particularly larger eels either, but this 8 to 15 centimeter stage, this elver stage. And I think it has to do with the glass eels are doing this transition from saltwater to freshwater, and their bodies, their muscle mass just isn't set up for ascending barriers. The older eels, they put on some mass, so the larger eels, but it's almost too much mass to crawl. There's something about that elver that is this sweet spot between musculature and mass that allows them to be really good climbers. And you've seen that again and again. So that elver stage becomes a very important stage when you want to think about access over dams and over barriers. You can kind of fine-tune your efforts to target that size and age of eels. All right. So that's a little bit on our glass eel elver research that we've been doing. Again, I want to put special thanks to Tom Lake and Bob Schmidt for the work they did in the Hudson, and in particularly Carol Hoffman and Jesse Hornstein for the work they're continuing to do to sort of be New York State sentinel site on eel research. So, if there's yes.
So in terms of the speed of the glass eels moving up the stream during the times that the tide is going against them, do you have a sense of what their strategy is to not be swept back? Do they hang out under sheltered areas, or what do they do?
We'll very often, sometimes even when we're going out to check these nets or install these nets, we'll kick over a rock or some gravel, and whew, you'll see glass eels scurry out of these rocks and in the gravel. So it is very likely that during unfavorable conditions, which could be high flow against the way they want to go, that they're sort of, all right, let's hunker down and let's wait for the tide or the current to change to help us out. Our further down river sites closer to the ocean show a fairly strong signature with spring tide. So full and new moons, we see higher levels of eels sort of using that to ride up these tributaries. But as we get further upstream, further from the ocean, that lunar signature tends to get blurred out. There's probably other things that are going on, other environmental factors. You don't see that strong signature when we get up, let's say, north of the highlands, for example. All right, one last question, then we'll go to the next speaker.
Question, comment about the age of eels. And as a species that lives on water where it's sort of thermoregulated by the temperature of the water itself, particle age perhaps isn't an ideal measure of age. Because I'm assuming upriver the temperature is going to vary both hotter and colder, and both of those can have significant physiological impacts on the eel.
They absolutely do, and in fact, what I don't have this in a graph, but I can show you afterwards. We've seen pretty good indicators of a temperature threshold. So we do not see a lot of glass eels coming into tributaries when the water is less than 45 degrees Fahrenheit. That seems to be a threshold. Once the waters warm up, there's not a correlation, higher temperatures equals higher eels. It's more of a threshold that won't they come in, and if it's colder than that, we are really not going to see a lot of glass eels. So we use that as one of our indicators to sample.
So they survive in that water during the winter.
They survive, correct, that's right. They do survive in the water during the winter. As do a lot of other tributary fish species. Yeah. So we've covered the overall life cycle. We've gotten through the leptocephali, the glass eel, the elver. We're hinting at this yellow eel stage, the stage that most fisher folk are aware of, that we've seen. So let's continue this life cycle research a little bit, and let's get into sort of our Karin's going to come back and tell us, okay, the eels are a little bit older. What do we know about these guys?
Okay. I'm not going to tell you all that much about them, but I do want to follow up on your comment about overwintering. I was at a conference which was themed, it was a chapter of the American Fisheries Society, it was themed on diagonal fishes, migratory sea-run fishes. And there was a guy from somewhere in Virginia, I think, who was a grad student, who had put tracking tags on eels. And they were kind of expensive tags, and one of them got lost in the wintertime, and his advisor forced him to go find it because it was expensive. And what he did was, so he took it long and sort of went all over the place until he found a signal, and the signal was in the creek bank, and he had to dig with a shovel, and he found that this eel was up, it was hibernating essentially up in where a tree root had died, and there was a big macropolar hole. This thing had crawled up inside the bank and was asleep there, and to its surprise, it was found by this grad student who took the collar. So, okay, so I'm not going to tell you huge amounts of things, but I do want to get back to this otolith stuff because that's what I do a lot of. And so I wanted to talk about some stuff that we've been doing that's a little bit more recent. One of the things that we did was we put artificial tags into eels in our focal study river, which was the Indian Kill next to the Hudson River National Estuarine Research Reserve. And Sarah and Chris have been monitoring those populations steadily since the beginning of our project. But besides these artificial tags that we can implant in eels, and this is a picture up in the upper right of a yellow phase eel, and here's this eel wondering what its ID was, I guess, with the artificial tag. But the otoliths also serve us for many, many species of fish as kind of natural tags. The reason for that is because, and here's a little bit of a diagram showing you something about eels. So you can see on the upper right, there's the eye of a fish and the gills, and there's the brain case and the semicircular canals. And underneath those semicircular canals are these specialized little sacs which contain the ear stones. And those ear stones, the growth rate is regulated and they grow, as you saw, incrementally. In fact, in young fish, we can actually count daily growth increments when they're growing pretty fast. But these things are made of calcium carbonate, so they're not really like bone. Bone is apatite. It's a phosphate-based mineral. These are calcium carbonate in a form called aragonite, and that form, which is a little bit different than calcite, actually will entrain certain kinds of trace elements. And then they also have different types of isotopes in them, and people do a lot of work on that. So isotopes are basically the same. If you think of one element, it has different numbers of neutrons, and that kind of work is done a lot by everybody from geochemists to physicists and biologists, looking at those as ways of asking questions of science. So we can use these properties and look at things throughout the periodical table and ask questions about migration in the context of fish. When they're odorless, we can ask questions about migration, life history, but also provenance of fish. Where do they come from originally? And our own Dr. Waldman in the back there wrote the first, or edited the first edition of this book. You're not on the second, you're not the lead editor on the second edition. But get John's version, you'll see his name on it, the first edition. But one thing that people have been wondering about is whether or not we could use these methods on the American eel in the Hudson River. So as part of Sarah's thesis work, she did some age work on otoliths of eels. We sampled a few. The problem with otoliths is, although they're great for investigating many aspects of fish biology, you do have to sacrifice the fish, so we don't like to do too much of it. But this is one of Sarah's very nice preps, and you can see those nice increments. If you look closely, here's that sort of dark core, but outside that, she's put a black mark. That's the beginning of continental age, and then she's put white dots to note the other increments beyond that. But what we also did was we went down to our chemistry lab, and we have an instrument down there that allows us to take a very fine laser and take a transect from the core of the otolith all the way through anywhere we want. But basically what we do is we go along a major growth axis, and we analyze a suite of trace elements, and also things like calcium, which is almost 40% of the composition of the otolith, and we take the ratio of those elements to calcium. And also I'm interested in magnesium too, for various reasons, and we had that too. And so what I'm going to show you then is that we're kind of in the midst of doing some multivariate analysis of this work, but we're trying to ask the question of, can we tell where an eel in the Hudson comes from if it's in a tributary? Can we locate them back? Can we say that they have a provenance signature? And the answer is, although it looks horrible, is we can actually do it. This is called a canonical plot, and it's taking all those different trace element to calcium ratios plus the magnesium isotope ratios, and throwing them into an analysis and spitting out kind of a couple of canonical axes. And what you see here is little asterisks which mark the location of an individual fish in this sort of statistical space, but next to it is the label of the stream that it comes from, the tributary that it was caught in. So we collected them from 6 tribs up and down the Hudson. Minnesota is down in the estuary. It's pretty far down. The Blocky Kill, which is up here, is our furthest up. Remember how far up it is? Near Castleton. I'm sorry, I don't have the river kilometers on the tip of my tongue. But what I've done here, so those are the actual individual observations. And then what I've done is just located the average value for the different tributaries, kind of where those would fall out. And so we have Indian Kill, Cromilbo, which is right next to the Indian Kill, Fall Kill, which is in Poughkeepsie, not too far away, the Blocky Kill up north, the Minnesota in the south, and then the Black Creek over on the west side. That's how they fall out. But interestingly enough, when you run them through a classifier, all 53 of these fish, to my great surprise, came out 100% correct. Now, all we're doing here is we're taking the very outer part of the chemistry, the very outer part of the otolith, and analyzing that part of the chemistry, and asking, well, is that enough information to tell us where those fish came from, since they were caught in those streams? And the answer is, surprisingly to me, yes, we can do it. And so, and this is actually not so many individuals. If we bumped up the numbers, we would get much even better resolution. But even so, I think all of these were significantly different from one another, except for Indian Kill and Crum Elbow, which are right next to each other. So the geology of those two systems is probably pretty similar. So this is actually very good news for us. And if we can do it for eel, we can probably do it for other species as well. So that's kind of a little bit of work in progress on that. And I think at that point, I'm going to transition over to Sarah Mount. Yes.
So given,
You want me to go back?
so given that, and that was just on the outermost
Yeah.
layer,
Mm-hmm.
would we then be able to also kind of trace back through time and
Yes,
figure out?
in fact, an excellent question, and I'm not going to show you tons of data, but if you were to look at, let's say we're looking here at a transect that we make through, so we have the otolith cross section that you saw, right? So you've got the core, you got those things, right? So the core is here. What we see if we're looking at strontium to calcium ratios, what we would see is a giant peak right there. If we hit the core just right when we polish it down, we would see a big peak like that. And then if they hit the estuary, we're just going to see kind of something like that. And so the strontium signature from Sargasso is huge, and that we see in European eels as well. I wish I had a different color of ink. But if we were to look at a different element, manganese, if we were to look at manganese to calcium ratios, in a lot of these eels, we're actually seeing as they come off of this big, they're coming out of the sea and hitting the estuary, maybe even before they drop down to this very low strontium value, we're seeing something like in manganese, something like 6, these little twin peaks here. And then we may taper off. Sometimes they get high again. And I think what's going on here is they're going through some, they're perhaps going through either some wetlands with highly reduced sediment, or possibly some water that's got low oxygen. And that's another thing I've worked on quite a bit, is looking at various properties of hypoxia, of low oxygen water. And Dr. Jackman over here has done some of this work too. So flame goes around, right? And I won't talk about it now, but there seem to be other things in here that tell us that maybe some of these eels have very particular ways of coming up. So the small gills seem to have a unique pattern in the few eels that we've looked at so far. Something's going on that suggests that the way they're getting up through the river is character to the bend. So as we work through more of the data, we'll hopefully be able to see it. We can test that. So next, then, I'm going to pass the baton on to Sarah Mount. One more quick question, yes?
I'm just curious, if the tributaries have distinct families of eels, could you say that the same eels go back?
That's a very good question. I don't think we can say. That's way beyond our abilities. And the only thing I can say is that we know that eels are what's called pan-mythic. If you try to look at their genetic composition and look for differences, there is none. There's absolutely none. People look at with their fine tools as they can, and they cannot find differences between American eels within that species, European eels within that species. The only place you see differentiation is there's a hybrid zone in Greenland and ice. But having said that, we know that genes express in different ways, and people up in Canada are trying to unravel some of that now. But I don't know.
That would be one, right?
That would be amazing. We could do that. Maybe we win the Yale Nobel at that point. So I'm going to ask the whole question so I can continue and onward to silver eels. But I just want to note this was the largest eel we ever caught. It was bigger than our measuring board, and it was a nice one.
All right, so I'm going to transition to talking about the migratory, mature life stage, the silver eel life stage, and really the transition from the yellow eel that's living in freshwater to the silver eel that's going to make this journey from the land back out to the Sargasso Sea, where they spawn once and then die. And we say that eels spawn in the Sargasso Sea, but we don't actually know the specific spot in the Sargasso. And the way that we know that eels spawn in the Sargasso is from the work of looking at leptocephali. So that's where they find the smallest larval eels. No scientist has ever seen adult eels spawning out in the Sargasso Sea. There's still a lot of mystery to this life stage, which is one of the reasons why it's an interesting life stage to study. There's still a lot that we don't know. There have been some groups that have tried to track silver eels on their way from land out to the ocean. And the way they did that was using these pop-up satellite tags. You can see the tags are pretty large, and a lot of it is float, because the way these tags work is you attach them to the eel and you program them to pop off at a certain time, and tags float up to the surface of the water and then beams the data to a satellite, packages it in an email to the researcher who set it up. So you get all this information of the location of the eel, the temperature of the water, the salinity, and the depth. So you can get a lot of good information about where this eel has been, the track that it's been taken. So they did this study up in Canada, and they tagged, I think it was 38 eels with these pop-up satellite tags, and of all those eels tagged, one individual, one female, was successfully tracked to the edge of the spawning ground. So on this map here, this box, it would be the supposed spawning ground. So they caught the eels in the St. Lawrence. They gave them a little bit of a headway and released them off the coast of Nova Scotia, because in the beginning of the study, they found a lot of eels were being eaten in that journey between the ocean and in the river where they were caught. Now, usually, when you're doing scientific research, you want to have replicates, have more than one individual to make a conclusion. But in this case, because no one has ever seen eels spawning in the wild, this was huge eel news. This paper just came out at the end of 2015. So in the nerdy eel world, this is a really big deal. They were able to successfully track one individual to not even all the way to the spawning ground, but to almost the way. And so there's a lot of cool things that we can learn about their migration from this graph. What you're looking at is the red, the coloration is the number of days that it took. So if you look up at the top, you can see that it was 45 days from release to the northern edge of the Sargasso. And that's with this giant tag on them, which is pretty impressive that they moved that fast. And you can learn some things about the track that they take, which seems to be very direct. You can even see she was like, oh, I went a little bit too far. Let me hook back around, and then went straight south down to the Sargasso. So I like to tell this story just to show some background about how little we know about this migratory ocean-going life stage. So in our study, we were trying to understand a little bit more about the transition from yellow to silver eel, to try to get at some of that mystery, but in a place where we can study them more in the river and the streams that feed into the river. So we want, yeah.
Was the eel rising to the surface so they could get those measurements, satellite measurements?
The tag releases off of the eel.
So it's stored, the data are stored, and then when the tag releases, it goes up to the balloon takes it up to the surface, and then it feeds to the satellite.
Well, what's the site? I give it back up.
That's done. The eel may have gone on, but the tag
The tag release. That's the end of the data.
But they
Yeah.
used to be multiple.
It's all archived, it's data storage.
Oh, I got that.
So these tags, which are rather expensive, actually archive data on salinity, temperature, and depth, and I forgot something else so that they can get the location as well. They can calculate location.
Yep. So in this study, we're trying to get to that transition between freshwater resident yellow eel to a migratory silver eel. And one of the goals of the study was to develop a non-lethal index of maturity, of silvering for eels. You can see just by looking at them externally whether or not it's a silver individual. And that's so that that index can be used in future studies because there's so much we don't know about this life stage. We wanted to be able to provide a research tool for the silver eel. So the way we went about doing this was we did a combination of electrofishing surveys in the summertime to catch yellow and potentially silvering eels. And then in the fall, we spike netted to catch the mature silver eels on their way out. And we would put out this net, so it's the same kind of net we used for the glass eel surveys, but larger and facing the other direction. Now the eels are leaving the rivers and going out to the ocean, whereas the glass eels are coming up the stream to stay there for many years growing up. So we would pick rainy nights in the fall, which is when silver eels are known to migrate, and we cover the entire width of one site. Our main study site was the Indian Kill, which is right near the home of the Hudson River National Estuary Research Reserve, but we also chose that site because it's a small watershed. We covered the whole width of the stream because we want to be catching all the eels on their way out. And it's a unique stream in that you actually have a lot of habitat that you can sample before you reach a barrier. So we talk about barriers to migration, and in the Hudson Valley, most tributaries have one or several barriers very close to the main river. So this is one where we actually had a lot of sampleable stream before you hit your first barrier. That's the Indian Kill right here, and then we also sampled all those other streams that Karin mentioned before in her OLS study. And on any eel that was bigger than 25 centimeters, we took a suite of external measurements that were specifically chosen to look at indicators of that silvering process. So like Karin mentioned in the very beginning of the presentation, as they're maturing, they go through a set of physiological changes. Their eyes get larger so they can see in the big, deep open ocean. Their fins get a little longer so they can swim, make that journey. Their head shape may change a little bit, but the ones that I'm going to focus on is the eye and the pectoral fin. Those are the markers we found to be the most significant, but also size is big one. So we looked at total length and weight. And then we took all of the individuals, almost 1,000 that we measured all this data on, and put it into a cluster analysis to break out into different maturity groupings. And we found significantly different clusters that fit with our knowledge of eels that we have from other studies. So we were able to say that some clusters were silver females, which are bigger than silver males, because we know that female eels are much bigger than males, especially at maturity. And then we can look at the yellow eels, which are, again, the freshwater residents, and you can see that this one is just yellow, which means they could be male or female. Can't tell. And these ones were just big enough that we knew they were females. Yes?
How were you able to assess them?
This is just based on size. So once you get over a certain threshold, 400 millimeters to 450, it's too big to be a male. So that's why these are definitely females. And we did not sex them. So that's why this one, we don't know what this yellow cluster is. It's probably a combination of the two. Yeah, you have to kill them to do that. So the whole idea of this is to not do lethal sampling as much as you could. Then when you look at some of these key maturity indicators, if you look at the Pankhurst eye index, which is an index of the eye diameter based on the length of the eel, you can see that there's a clear difference between the mature eels, which is the kind of gray colors, and the immature yellow eels, in the yellow color. And that line there is the threshold from the Pankhurst index, which is at 6.5, where Pankhurst separated out mature and immature eels. So our analysis fits in really nicely with what Pankhurst said it should. And the same is true of the fin index, that's the pectoral fin, the one right by the face, where you can see the mature eels have larger fins than the immature eels. That was nice that that happened. And then we did discriminant analysis to come up with functions to classify new individuals
into these groupings. And it's not at 100% like the odorless analysis, but it could correctly classify individuals to 83% overall, which is actually pretty good and very comparable to other indicators of this kind of thing. And when you do field science, 83% classification rate is actually very good. So now if you capture an eel and you take those suite of external measurements, you can plop it into a model, and it'll tell you which maturity stage it's at from this analysis. So on the Indian Hill, which is our main sampling site, we also tagged and recaptured individuals. This is using pit tags, so small 9mm personal integrated transponder tags. And we are able to learn some things about eel growth, movement, and maturity. So far we tagged about 350 eels and recaptured over 100 of them at least once and many multiple times. So you can see over time on an individual eel level how much they're growing, where they're going, and how they're maturing. Overall, we see that eels in the Indian Hill are growing about a centimeter a year, which fits well with other studies that they found on growth. But again, very variable. The magic word for eels is variability, of course. And then 62% of them don't move, which means that they're always caught in the same exact reach where we caught and tagged them. And even more of the ones that did move, a lot of them moved to just a neighboring reach, the one right next to it. So eels move a whole lot from the Sargasso up into these freshwater environments, and then they stay somewhere. And then they move a whole lot again to go out to the Sargasso and so on.
Is that going to define reach?
So a reach is just about access for us, but there's no barrier separating things. So when I say neighboring reach, it's really just
200 yards.
Yeah.
200 meters in this case.
And then for maturity, to show that, I'm going to just tell a little story of one eel that we caught and saw it maturing over time. So this map shows our sampling stream for folks who have been to the Norrie Point Environmental Center. We're like just down here. And this is the Indian Keel. There's a little marsh at the mouth. And this eel we caught in July, and it had an eye diameter of 4 millimeters by 4 millimeters and a fin length of 14 millimeters, which is all fine and good and normal. And then later that year, in October, we caught that same individual because we had a tag in it. In this A site is in the spike net in the fall. So because it was October and at this site, it was spike netted, which is specifically sampling to catch eels on their way out. And that individual had a bigger eye diameter and a bigger pectoral fin. So we can tell from that data that that eel was on its way out heading to the Sargasso. The timing is right, and it had grown in those two key indicators of maturity. So that's just one story, but we can see this in other individuals as well over time. And when you look at all of our samples at all the eels caught in the Indian Hill overall, you can see the same kind of story, that you can see the percentages of the number of eels in these different maturity groupings. So again, gray is mature, and yellow is immature. You can see the silver eels increasing as it gets into that fall migration season. So they come in the spring, and then they leave on a rainy night in the fall. So just to summarize that silver eel research piece, because there's so much that we don't know about this life stage, research tools are going to be really important in the future of understanding how this life stage and really how the whole species works. So potentially some uses for this new maturity index could be looking at productivity in a given system and then comparing between different habitat types, which ones are more productive than others. So there's really all kinds of different things you can look at using this new tool. And that's all that I'm going to pass it back to Bowser with the trick photo, unless there's any further questions. Yes.
I'm just wondering if you know, like, on average, how long each cycle lasts?
Well, it's very variable. It's very variable. And it also depends on where the eel is living, food availability, habitat in that place, and then whether it's male or female, on how big it's going to be. So females need to get much larger, and they live longer than males. there's a whole lot of things that would, for,
I can tell you that the English eel that I know is over 100 years old, well documented.
but that would be a rarity, I would say.
Yeah, there's not many of these well documented.
Although, who knows?
Yeah, that's true.
No, this is in Sweden.
So European eel.
Yeah.
But I found ones with like 20, 25 in the ones in our transition district. Yeah.
How long was that 100-year-old?
Oh, well, the ones, so, these eels, I think there've been a couple of cases of well-studied eels. They tend to get rather a lot like little old men. You know, they're just kind of choking up after a while. The ones that, my colleague Håkan Wikström in Sweden, was on a TV show, the TV show was sort of about interesting things in nature, and it was just, that show was about very old animals, and, but he crawled with the MC of the show, he crawled down a well where this eel had been put in the 1800s, I think, and had been fished out once to check it in the 1930s, and then they decided to go back in to see if they could find it. And my colleague had a small net that he made, and they crawled down this well with flashlights and stuff, and sure enough, they finally caught this thing. It wasn't very big. I mean, they used to throw eels down wells to clean them from vermin, is what they did. So this poor little eel was, you know, 120 years old or something. Yeah.
So we're gonna wrap up with a little bit of a dip, a little glass half empty, and then we're gonna end with that glass half full. Don't worry. An article a couple of years ago really hit me hard, and it was written by Dr. Waldman and Dr. Limburg, and I'm going to paraphrase it, and it was basically telling us that society is losing touch with its migratory fish, right? A lot of our migratory fish are academic topics now. They used to be culinary topics. We used to catch them and eat them and see them and experience. I mean, we as the river societies of the world were in touch with our shad and our sturgeon and our spread bass. In a large part, we have lost a lot of that. And it's a chicken and egg thing. Did we lose the attitude because we lost the species, or are we losing the species because we lost the attitude? And I would say, and that article hits me hard. Everybody should go look that up. I want to say it was Gale 360 was the article. But it's
a bioscience.
A bioscience, that's right, bioscience, sorry. But here's the glass half full. I really do truly think that eels are a turning point that allows us to take that attitude and change that. It is this beautiful, beautiful species. And in our original outline, it says, you know, evolution from a study object, the beautiful academic interest in eels, to a river icon that brings people together. It is, first of all, think about it. Eels are found in every habitat that we have in this Hudson River, Raritan, watershed, New York Harbor group that we have. I mean, New York City, Albany, little watershed ponds, you're going to have eels. So it literally, that interconnectedness that's a little bit abstract, it's not abstract for eels, it is life for eels. They are ecology, persona, or animalified. And there's just something about them that is compelling. Hello, they're cute. They're accessible. You can find them, and they're usually miraculous. You know, this guy took this picture, geez, not even a week ago, and this girl, who's a Poughkeepsie High School student, she came down to the ball game for the first time, had no idea what was going to happen, had no idea that her creek had this in there. It was a little bridge to that connection for her. It was an eye-opening experience for her, and we like to think that eels can be an eye-opening experience for everybody, not just about eels, but about the whole watershed. There has been, luckily, a slow but incrementally growing attention on stream connectivity that I think eels are a part of. And citizen and community science efforts like the Eel Project are sort of trickling this constituency, the mycorrhizae that is getting people interested in things like breaking down dams. Here's a dam on the Wappingers Creek in Dutchess County called the Shat Pond Dam. was definitely not doing anything for stream connectivity, and really, frankly, wasn't doing its job as a dam. And luckily, with some grant funding, that dam, as of last summer, is no longer there. So there you've got one more piece of stream connectivity. New York has got a long way to go, but darn it, if we aren't taking those first steps at places like Chat Pond Dam, or looking at the Bronx River, and how can we increase connectivity there, or all throughout the watershed. There's a real piece to that. And I think that efforts, both academic and community, are helping to bring that sort of recognition there. It's not too late. The eels may be that fulcrum that can get us back on track, focused on diagenous species in a way that maybe we've lost for a while. And things like getting volunteers and students out there in waders, hey, look, I'm in a stream, I'm figuring this out. That is that first little step to have that done. I'm really happy about this crowd right here, by the way, because this is a great crowd where we have gotten some phenomenal, top-notch academic researchers. I mean, the cream of the crop in here. But we've also got a strong showing of education efforts, like the science bars, like river projects, like the Mystic Whaler crew that's here. So you've got this perfect bridge. And I want to give a particular shout out to that new generation of educators. All right, I'm talking about people like Dylan and Kate and Jackie, who, I don't know about you guys, but I remember when I first started as an environmental educator, every time I would get together with scientists, I would have, oh geez, I'm a bit of a pretender here. I'm doing the best I can. Is it a bivalve or a gastropod? I got to figure this out on the fly, right? And one of the things that a group like this, the Hudson River Foundation, can do is bring those parties together, and particularly for educators who are starting out in their careers. What you're doing is tremendously important, not just for the kids, but for the eels and for the researchers. By building this constituency, it is absolutely helping out that whole ecology. It's absolutely helping out that whole zone. So even on those days where, geez, I have any effect at all? Absolutely you did. And the two volunteer quotes. I like this. This is like, you know, you put this in your grant application, right? There is life against all odds in places where you would think there is nothing. Interest and opportunities exist in the scientific community for amateur naturalists to make a contribution, right? Oh my gosh, put that as the front piece to a journal article, all right, in Citizen Science. And then I also, it doesn't have to be so grand and mighty. I like this high school students line as well. At first, I only did it for extra credit, but then I realized it was cool. You know what? Keep it accessible. We'll take anybody to come down, take a look at those eels, and really help to build that constituency to start turning this corner. All three of us, Sarah, Karin, and I, want to thank everybody for putting up with this very long presentation. It has to be long. It's a species that deserves some time. And it's a species that we still have so many questions on. It's still ripe for research. And we just have some extra thanks. Also to Mr. Bob Schmidt, who couldn't be with us in person, but we still love him. That's for you, Bob. And if there are any questions, I'm sure that we can take them. Thank you all very much. Sorry to leave the show.
I just want to make a comment because, I'm from the river, working with this wonderful group of high school students this year. We do it, but they show up at the show. Gotta go over there. And then it is such an amazing thing to pull up the inbox because they bring people into the community. They have, you have to show them that people walk by and last year. So I compliment all you guys on your work. It's a wonderful program. And you want to do it.
I have a question for you about the eel moss. When you pull them up, are the glass eels adhering to the filaments, or they're dripping off, dropping off?
Both. When we pull them up, an eel moss, we make it out of 5 feet.
Look at this joint right in here. Basically, they like to wind themselves in here. we suspect their instinct is not that, when this gets disturbed, it's not, let's leave. It's, oh shoot, I better burrow in a little bit deeper. So then you have to sort of shake and squish those eels out of here. And we follow some protocols that were really developed by Ken Abel and Michael Sullivan. I might have Sullivan wrong, sorry about that, in New Jersey, or Rutgers and others. So that's an eel minus its big concrete disc to weigh it down. So it's pretty beautiful.
So then you count the ones that fall into the bin or whatever.
Yeah.
Whatever you.
Yeah.
We pour them into an aquarium net, and then we empty the aquarium net into a stainless steel tray we have, and then we count them. So it's interesting, we're getting crayfish quite a bit with them this year. So it seems to be an increase in crayfish.
We're going to be trying to deploy this one up in the Mohawk in a couple of weeks. See if we can see anything up there.
It's great team building, separating the rope into the strands, and then when we're on the water, when it's tight, we have little sewing circles or whatever, we untwist the braid. We just show that wonderful technique at a conference with Chris, where we have a whole room unraveling.
Team building, that's what I call it.
Comparing ratios.
Haven't seen any cesium or cobalt, or haven't seen anything like that. No.
What would be the tributary around it?
Pardon?
What should be the area that would closest to that?
Probably Deep Steel Hollow or something like that, which would be an example. But I'm really curious about things like uranium, whether that's taken up. I actually analyzed some yellow perch otoliths from fish that were caught outside of a nuke in Lake Erie. No uranium. I don't know. I'm still research. Yeah.
To piggyback off that, would the levels of manganese to calcium where those spikes are, is that just every eel that you sampled, or specifically the Hudson?
Not all.
Because given that Indian Point is using the river to cool their rods.
Yeah, that's probably not. This is probably downriver. This is probably just entering the Hudson. Yeah. And just to orient you guys, this is supposed to represent kind of this transect through here. So that's kind of the core right in there. And then I could show you other elements that would also indicate that this fish was passing through a very strong estuary zone at this point. And then here we see some that show these little twin peaks of manganese and others that don't. And I don't know if my own hypothesis is maybe it's Limburg Bay. I don't know. What do you think, George? Could be. Yeah.
Karin, we have a couple of questions from the webinar.
Oh, okay. Yeah. Let's see.
Chris.
Okay.
Regarding the slideshow showing numbers of eels caught per day per year, was that data normalized to the number of sites involved in each year? I think you said it was a measure of eels caught per day per unit effort, but I wasn't sure. Yeah, I would say that it is normalized. It's basically, if you look at all the sites and all the days over all the sites, it was all of the eels caught divided by all of the days at all of the sites. So it's normalized in the sense that it's eels caught per day per site. So we'll have fewer eels, generally you would think, on the years where we had fewer sites, but hopefully the extrapolation holds true. So the quick answer is yes.
You want to take the second question up there? Sure. You want me to?
No, I can answer. So another question from online is, do we know the effects of the watershed on the populations of yellow and silver eels, and hence the numbers that are successfully going back out to sea? What I'm concerned about are the effects of habitat modification and legacy contaminants. So one thing that we did look at but didn't include in this presentation was parasite burden. Karin touched on a little bit about parasites and how that can impact the ocean-going phase, so the swim bladder parasite. And we know from that individual that was tagged and then tracked back out to the Sargasso that they make these huge migrations up and down the water column, and when they go out to the oceans, that's where it really had an impact. And we found some interesting results in this recent study that are kind of different from the one done in the early 2000s, but lens that in more forested watersheds, they have higher prevalence of the swing bladder parasite, whereas more urbanized and potentially impacted watersheds had lower impact. So there might be kind of differing, pros and cons for these different environments.
In fact, early on in the lens study, we found a little bit more parasitization in those urbanized parts of the system. So things just must be kind of dynamic, I guess. Hard to say. Yeah, you've been waiting a long time.
Would it be possible to determine the location of eel spawning grounds in Syracuse Sea by accumulation of otoliths on seabed?
Wow. So the question was whether or not you could find this elephant graveyard of otoliths at the bottom of the Sargasso Sea. And you have to remember that these are about a few millimeters across. So I think the odds of finding that is smaller than a needle in a haystack, is my guess. I've actually thought about a way I would try to track eels to the Sargasso. I would put tags on white sharks and watch where they go, because they actually they like to prey on eels in the Sargasso. They move there. So I suggested this to somebody from some nature show in England, but I don't think they ever did. However, it's an idea out there.
To piggyback on that, one of the questions online was, what about the tag eels that were released that presumably go to Sargasso Sea? What happened to all the other 30 eels that didn't get caught? And there's strong evidence to suggest that they were probably eaten by sharks, and that is based on the fact that the signature on the tag basically went down at depth, stayed at depth, slightly warmer than ambient water temperature, which poor beagle sharks are able to, I don't want to say thermoregulate, but they just tune up their bodies a little bit more than ambient water until the tag was pooped out and then sent all this data out. So it's very likely that most of those 30 eels were eaten by sharks or some other big, tuna or another big predator.
The spawning grounds for the ox for eels spawning is pretty well known. The thing is, no one's actually seen it. People won't actually witness it. But picking up on the otolith idea, you could look for environmental DNA in the bottoms of the consumed spawning area. If these fish are spawning in time, they're decomposing the bottom, and there should be a patch of eel DNA that could be detected, and then an area where it can't be detected.
Do I sense we're going out to sea next to Noah?
I just create an image of Karin Limburg in like an old school diving bell, You're like a metal detector.
Submarine would work.
I heard anecdotally that many of the glass eels, which used to stream out of the Sargasso Sea, are now being intercepted by fisher folk with massive nets, and that the overall eel population is crashing because of the new kind of interest in eating the glass eels. And this was, just casually said to me in a hallway. So there may be no scientific evidence to it, but I wondered if you have any sense of this becoming like a new hot restaurant item and having an effect on them, particularly in the way that they're caught going through these conditions.
So I haven't, and it may be out there, I haven't heard of any studies or investigations of sort of intercept fisheries offshore, sort of trawling up eels with super duper fine trawl nets kind of thing. But I do know that of course there's interest in glass eels. When you have a commodity that's going for $2,000+ a pound, there's going to be interest. So other states, I'm sure are interested in this. I know that, for example, in Haiti and the Dominican Republic, they have increased their glass eel fishery efforts. So there is a lot of focus on this, but at the same time, there's also a recognition that this is a species that is not out of the imperiled woods yet in terms of overall populations. So, the American eels have been considered twice for the endangered and threatened species list.
So the U.S. has not declared them endangered, but the International Union for the Conservation of Nature has declared them endangered for this very reason, because of vulnerability to these fisheries. And I actually, John, you could comment on it too, but I think it would be highly inefficient to try to sieve them out of the ocean when you've got these concentration points where on a dark night, all you need is a headlamp and a bucket and a little dip net, and hopefully the conservation officers aren't around, you can make $50,000. So,
the New York State Department of Environmental Conservation does not endorse that situation.
John would be the one to ask, I guess. DNA, eel DNA.
Yeah, there are studies and they basically suggest mixing.
Yeah.
So there's a lot of variability, but it's not partitioned into geographic units.
The thing that seems most intriguing is the fact that they can actually express in different ways. So again, a lot of work's been done by these there's a couple of groups of Canadians. They put the tags on. They've also tried to do a lot of stocking. So they stocked, at one point, eels were taken from the Maritimes, the Elbers, and stocked into Lake Ontario. And when that happened, they became small male. And so there was something about that then they started looking at the DNA. And honestly, it's beyond my understanding of genetics. But there is apparently some very complex sequence that happens where they sort of settle, some trigger goes off and says you're going to become a short male or you're going to become a large female. And one of the things that we do know is that density typically creates the conditions that they're high density, that creates conditions for them to become male. So that was often discovered in aquaculture facilities where they were they're trying to get large eels, male things, and small eels because they were all crowded together. And one of the reasons, as I said, there's a conservation issue associated with getting eels further up, penetrating in further into watersheds, including the Great Lakes, including the Mississippi, including the far off cribs of the Hudson, including the Mohawk, is that the further they go, the more dispersed they are, and they tend to become female. So it is argued that the reservoir eel in the Great Lakes was the female component of the at-large American eel population, and the fact that that's been decimated through damming, for the most part, fishing and damming, and perhaps other unknown factors, means that we're really hitting the female component hard. And there's very little known about them in the Mississippi, for example, where we know they also go up. They used to go up into Iowa. They were caught at one point in Minneapolis. So far up, then the Adirondacks, so they historically could get around. And our colleague Dave Secor referred to this as the reservoir effect, that you have this huge storage of multiple generations of eels that we sort of picked apart, if you will.
Yeah. The other thing about harvesting eels is, unlike other species, if you catch a big 40-inch striped bass, well, that striped bass has probably had a chance to spawn multiple times in its life. With an eel, at any point in its life cycle that you remove it, that genetic diversity is lost. It spawns once. So whether you catch that as a glass eel or as a silver eel that's out migrating somewhere, it becomes a trickier topic to manage and to think about as a potential food resource, because it's a one-shot deal for the eel.
Sure. Chris, in the beginning you said the eel population seemed to be increasing.
Well, our last eel recruitment,
based on your surveys, but kind of normalizing that data like the other gentleman asked. Do you think they're fishing better and smarter now?
For the most part, no. We've actually...
Aren't you pulling nets on certain days where you wouldn't have known?
No, we have tried pretty faithfully to keep our nets where they are, basically place them in the same spot in the same tributaries year after year. We're pretty good about that. We will occasionally, if we remove a net for a couple days, it's usually for a high flow event. And honestly, on those days where we have left nets in, the catches have been low anyway. I'm sure that there may be some bumps in that, but I feel that our effort is very similar year after year in terms of placement, in terms of how we check the net. It's a very, very straightforward process. There's not much else in the net other than glass eels and elpers. And so there's only so many things we could change and screw up if we wanted to.
We're putting the nets in the same spot here.
Yeah, as streams allow. Remember, some of these streams actually change over the course of the year. So we'll have to move around gravel banks and just have the morphology of the stream itself changes. But we try very hard to replace that net at more or less the same spot every year. It's a little tricky with some of these streams, but there's not much else we could do.
Kathy? Travel and, did you say already, or do we know, could you say what determines what freshwater tributary they go into? If it's Annexia, and also when they're traveling down to the Sargasso, these vertical migrations that are so big. Oh, I think that's the answer. That's it? Okay, thank you. The vertical migration, thinking that they might sort of want to take a straight route, I'm assuming, is that like current related and drift related, these vertical they're getting out of or into a current that way, or is there something else about the predator avoidance?
Yeah, I think the big one is predator avoidance, to go up and down at different points of the day. And then the short answer to how they pick which tributaries to go into is that we don't know. That's the shortest answer. But it's probably based on some combination of environmental conditions and not that their parents were from a certain tributary. That's the answer. Yeah. But it probably has to do with the strength of the Gulf Stream, temperature indicators, the tide cycle, all these things combining. So it might seem a little random, but it's probably just so many factors coming in at the same time that it's hard to tease those differences out.
That's why I was sort of fascinated seeing that. I just happened to notice in Sarah's data that it looks like there may be a bit of a pattern for the Fall Kill, and you guys noticed, and so maybe that there are rivers within rivers. There may be some currents that guide the flow of eels up to the Fall Kill, and the same with the Hudson River.
Right. There's like river morphology you think about, river bottom, substrate.
Yeah, I mean, really, to jump back to George's question, is one of the things is we can't span the pike across the whole tributary, because then we would be cutting off access for other species to use. So it's part of it. And there may be that rivers within rivers, there may be little micro changes from season to season, that some years we hit the jackpot, some years that micro river is following the other bank. And it's very hard to, so many variables start to add up in that story.
Question in the back?
Yeah, a question for Chris on species. So I think you made a pretty good feel for eels as being a dynamic and acute species, and I think probably everyone in this room would share your sentiment. But I would almost expect an opposite reaction from students, especially the younger ones. So I was wondering if you'd seen that and how you sort of overcome that possible ugliness of the.
I call it the ooh factor, and almost the question was about the fact that are eels really charismatic, because it would seem that the opposite would be true. They're distinctly non-charismatic. And it's something that I call the ooh factor, which most people, when they're first exposed to eels, even the little baby eels, their reaction is ew, right? And then honestly, when you hold one of these in your hand and you can literally see their heartbeat, I mean, literally, you look at that ew changes to ooh, and that's honestly that's my job, and that's the job of many people in this room, is to overcome that societal instinct to abhor or disdain this slimy creature that looks like a snake or a worm or something. And you're right, they can be uncharismatic, but it's our job collectively, and everybody in the room's job, to sort of break that mold. And I think that this is one species where you can, and your gains are huge because you're starting so far down.
Yeah.
Going back to the signature chemical ratios, the different tributaries. What accounts for that? Is there something in the
local geology,
probably? Stream,
yeah, it's probably the local geological conditions there is what I would expect it to be. So to sit down more closely with it, with a geological map would be the thing to do. But what we typically try to do when we when we do these analyses, now we didn't do it in this case, but very often we'll try to build what we call a geochemical atlas, where we take water samples across the year in different places and try to see if there are some sorts of signatures that might be of interest. I've done that in Grand Canyon, for example. I've done that in parts of the Hudson for another project. But to really do that kind of a job for the whole watershed would be a fairly good-sized undertaking. Maybe when mass specs get for optical emission spectrometers become cheaper, we could have citizen science geochemistry. You know, it's kind of fun.
And on your the citizen science line is good. On a follow-up to your charisma question, the other thing is all of these questions that would come up. The other beauty about eels as a topic is it's very legitimate. This is not a species that we understand very well. So these aren't sort of like canned laboratory. Oh, let's look at fish. Oh, isn't that cute? Then let's let it go. This very simple data collection. Let's look at the net. This is actual real data that is of value. So there's an interesting legitimacy. And in that topic of citizen science, or what I kind of like to call community science, community-based science, finding these legitimate phenological often topics is a really important part of making these projects work. And then that's what that's what attracts scientists like Karin Limburg and John Waldman to pay attention to this cool thing that's going on, which adds to more studies, which adds to more legitimacy. And so finding those sweet spots of student appropriateness with scientific appropriateness is a really fun part of my job.
And then do one from the crowd.
Okay.
So we spoke about how, especially in the Hudson River, where the river will switch currents, the eels will kind of seek shelter for a little bit.
Maybe,
yeah. So given that in the case where they may not just be instinctive who, okay, swim upstream, swim up the current, and combining that with the data that the satellite tracker had, is there now any further research to kind of figure out what that mechanism is that an eel may use in order to be able to orient itself while in the water?
Oh, yeah, actually, there's a paper that just came out in Scientific. No, I guess it was a reportage, but I didn't read for Scientific American. The paper came out. Where did it come out? I can't remember now. Anyway, there was a paper that came out recently about baby eels. So we do know that many species of fish. The question was how do fish orient? How do they know where they are? And it has been known for a long time that many species of fish have magnetite in their brains, and they actually are sort of like they can detect geomagnetic anomalies. And so it's been demonstrated in the past that adult eels use some sort of what's called a magnetic map to orient, to find their way back to the shore. That's in fact maybe that straight line. The beeline might have been that. But now baby eels have also been demonstrated. Leptocephaly has been demonstrated to have the capability of doing that as well. So that paper just came out. I can show it to you if you want.
And that was magnetite?
Yeah. So magnetite is the likely mineral. Yeah.
And how about addressing one more? You guys
Sure.
Getting a little tired.
So you folks on the internet out there, this is the last one. How long do silver and gray mature eels remain in habitats where they live most of their life cycle before going back out to sea?
Highly variable.
But I think one of the biggest differences is between males and females for that one. So females, like I said, get to be much bigger and so they live longer, and you can use like around 20 to 30 years, but there's a huge amount of variability in there. And males are going to be much younger and smaller, so they could be 5 to 12 years old, but again, huge amount of variability in there. So it really depends.
And I know from working with European eel up in the Baltic Sea that those, the ages go much beyond that too.
Yes.
So it's not unusual to find a 40-year-old eel or sometimes a 60-year-old eel. So, you know, again, respect your older music.
Real or Elvis?
Well, that was a terrific marathon. Thanks for all of you for coming and those on the webinar, and keep looking at our website for future seminars. We're probably going to take a break for the summer and come back in the fall. And if any of you haven't filled out or an RSVP to come today, we have a sign-up sheet at the front desk. If you could leave your name and your email address so we can keep in touch with you, and we'll send out notices on future events. So thank you very much. You guys. Thank you.
Mark?